right, I think we're going to get started. Don't feel shy about moving closer to the front of the room. Anyways, we're very pleased to have Tae-Hee Lee visiting us today. Tae-Hee is a professor, assistant professor at Penn State University. He got his PhD at Georgia Tech, working with Rob Dixon, and he worked on various kinds of fluorescent tags for biomedical applications. Particularly, there's what Dixon's group was interested in are these tags that involve the use of silver clusters, and so that's been a fruitful area. And then after that, he was a postdoc with Steve Chu. I guess initially at Stanford and eventually moving to Lawrence Berkeley Lab. And with Steve, he got involved in single molecule studies of a variety of problems in molecular biology, and some of that work has since continued on in terms of what he's doing with Penn State, and he's going to be telling us about that today. And title of his talk is Single Molecule Studies on the Structure and Assembly of Nucleosomes.
All right, thank you very much, George, for inviting me and giving me an opportunity to present my work here. It's been a fun day to hear a lot of interesting sciences going on here. So my turn to return the favor. So I'm going to talk about mainly the single molecule studies of nucleosomal structures and the assembly and how these are implicated in the mechanism of gene regulation activities. The main theme of my research program is basically single molecule structural biology of macromolecular assemblies. The reason that we focus on mostly macromolecular assemblies is because it's slow, big, so that's amenable for single molecule methods, especially fluorescence methods. You have only tens of kilohertz of photon emission rates. If you wanted to look at something between these photon arrivals, there's really low chance only to detect any events going on between these single photon events. So that's mainly the reason that why we focus on macromolecular assemblies. And they are also dynamic, so it's a very interesting problem from the single molecule perspective. This is one heterogeneity that we can deconvolve by our measurements, structural or conformational heterogeneity. If the energy barrier between these conformations low, you can see the dynamic behavior. One more interesting and important dynamics of biological molecules is rare events that could be pretty important in their functions in cells. So these three types of examples will be discussed today. The project that I'm going to talk about today is nucleosome structure and dynamics, but I have other projects also involving macromolecular assemblies, ribosome, and some DNA replication problems that we solve with collaboration with Sven Bankovic group in the department. Doing so, we constantly refine or revise single molecule methods to address problems and concerns raised by reviewers of papers and proposals. So I'll introduce the problem to you. Chromatin is dynamic packaging structure of eukaryotic genome, which is made of nucleosome core particle joined by linker DNA, and that forms this beads on a string structure and compact into 30 nanometer fiber and further compacted eventually into chromosome. Let me see if this is a movie showing how DNA is compacted in eukaryotic cells or nucleus. First, some histone complex called tetramer H3H4 tetramer binds, and then H2H to be dimer binds to form a core particle or the basic repeat unit of chromosome. That's nucleosome core particle that looks like this, and then further compacted into this. DNA in nucleosome core particle has about 147 base pair length, and its super helix that has 1.67 left-handed supercoil, and it's overly twisted. Jeff Hayes and Alan Wolf found that when DNA is complex with the histone, it has DNA helical repeat length decreased to 10.2 from 10.45 in free solution. So that's overly twisted structure of DNA. This is structural motif of core histones that wraps the DNA. Major alpha helix surrounded by two small helices, I omitted N-terminus regions and C-terminus regions. And these are the major helices that forms the octamer conformation of the histone core. I'll briefly go through the structural motif of the histone octamer core. First, H3 and H4 histone core forms helical handshaking to form the dimer, heterodimer. H3, H4 forms 4 helix bundle, 1, 2, 3, 4 helix bundles to form H3 and H3 dimer. And that together forms H3, H4, 2 tetramer. So when I talk about tetramer in this talk, that means H3, H4 tetramer, this structure. Similarly, H2A, H2B also forms this handshaking dimer structure, H2A, H2B. And H2B and H4 also forms 1, 2, 3, 4 tetrahelix or 4 helix bundle. That's the major motif of octamer formation. It's known that this tetramer also forms something very similar to nucleosome or octamer, but called tetrazome. And this structure is known to be stable at least in vitro. And add one H2A, H2B dimer, another to the backside of it completes the structure of octamer core of histones. And somehow Luger and Richmond started use this color code for H4 green, H3 blue, and it turns out that everybody uses this color code. So, so these colors are not random. It was first published by Luger and Richmond, 97 in Nature. So I followed that color code to show the histone core structure. And the interactions between DNA and histones crystallized in 97 and afterwards, a couple more seminal crystal structures indicated that these minor groups have contact with histone link between these helices that's rich with arginine and lysine. That's very basic. Or at least serine and threonine, that's polar. So these interactions between minor group motifs of the DNA and some of these histone tails, either tails or in the link region between the helices, that turns out to be every 10 base pairs periodicity. The other strand similarly interact with these red-marked basic residues, and then combined together, these residues form this major helix facing toward the histone core, interact with these basic residues of this histone core. So that's kind of a 10 base pair periodicity that was identified with them, and afterwards try to figure out what are the motifs that strongly position nucleosomes in cells. So how tightly the genes are packed in chromatin? That's about 50 miles of string packaged in a basketball. That's a high compaction, high degree of compaction. And this is what happens when genes are to be activated or transcribed. The TATA binding protein first bind to TATA sequence and then start assemble the initiation complex, and then with the activator touching the complex, it starts synthesize the mRNA or RNA. And this happens at least at a speed of 20, 30 base per second. So it's pretty fast process. And it's not surprising that not all genes are accessible this way at once. So only a portion or fraction of genome produces RNA at a given moment. And some genes are completely silenced at a developmental stage, so no polymerases or factors can access to these regions of genes. So there must be some active mechanism to dynamically control the gene accessibility or the activity of genes in chromatin or chromosomes. So that's the main problem. There are some mechanisms that governs this process, and one major mechanism is by modifying the chromatin chemically, and those are called epigenetic modification. Those modifications are mediated by enzymes, and some of them are inheritable. So especially the DNA methylation is inheritable. People started using the term epigenetics. It's on top of another layer of information on the top of the sequence of DNA. And recently, this term is being more widely used for all the other modifications on histone, core histones. So lysine acetylation, lysine methylation, arginine methylation, all these modifications seem to write some form of information that regulates the gene activity on the top of the sequence of DNA. So these epigenetic marks may play major roles in the gene regulation in eukaryotic cells. Two groups of hypotheses to explain the mechanisms of gene regulation by epigenetic marks is the first one, modifications act as signal for vector binding. So it recruits proteins that'll do some functions afterwards. So that seems to be fine with some modifications, but some other modifications have really weak binding proteins only, for instance, acetylations of histones and methylations of histone moieties don't really have well-defined strong binding motifs. Those chromodomains, bromodomains have really weak binding, only micromolar level at top, mostly millimolar level. So it doesn't really explain how this could be a part of the structure or the element of the structure. And there's another group of hypotheses to explain the mechanism of gene regulation by these epigenetic marks, which is maybe these modifications modify the physical properties or mechanical properties of DNA and histones and chromatin. So the accessibility of genes upon these modifications may be altered physically. Problem, they have tried to address this hypothesis, but the changes they've seen in an ensemble level was too small to explain the mechanism of gene regulation by this route. And our hypothesis is if the modification is globally conserved and if the modification is not structurally significant, such as methylation and acetylation, maybe it has higher chance to work in the second hypothesis, that's maybe the physical properties of the nucleosomes and chromatin be altered and eventually contribute to the regulation of gene accessibility. So we picked for that purpose DNA methylation and histone acetylation to test this hypothesis. DNA methylation in mammalian cells happens mostly at the C5 position of cytosine by methyltransferases. It's associated with mostly repressive chromatin structural states. The acetylation of histones happens at numerous different sites. There are 4 different sites at H4, that's N-terminal tail, H3, another 4, there's one more inside. So there are at least 14, 15 different acetylation sites on histone core, that's mostly in the N-terminal tail, lysins. That's done by histone acetyltransferases, HAT. NuA4 is a histone acetyltransferase that acetylates H4 and H2A. Saga complex acetylates H4, H3, and H2B. And it is typically associated with gene activation, although there is a modification or histone acetylation that's involved in suppression of genes. But mostly when you talk about histone acetylation, that means the genes activated in that region. All right, so CpG methylation, when I talk about DNA methylation, that's basically CpG methylation in mammalian cells. It's associated with gene silencing and repression. When you look at the appearance of CG sequences in human genes, the appearance or the frequency of the appearance is much lower than the statistical average you can calculate. But then there are regions where you find much higher frequency of appearance of CG dinucleotide sequence, and those regions are defined as CpG islands and happen to be abundant in promoter regions. So about 70% of human genes have these CpG island regions, and when they are methylated, it turns out that these genes are turned off or silenced completely. And especially in cancer cell lines, if you look at the tumor suppressor genes, the chances the promoter region is heavily methylated. And it's also associated with some mechanisms to turn off a set of genes completely throughout the cell's life cycle, X chromosome inactivation and genomic imprinting to balance the number of proteins or the concentration of protein produced from genes either from mother or father. So those mechanisms also have something to do with the methylation. Histone acetylation typically involves activation of genes. So the abnormality in histone acetylation is associated with typically the various developmental and proliferative disorders. So our hypothesis for CpG methylation is it may induce tight and compact nucleosome structure that may make the structure physically difficult for the polymerases to penetrate into the genes. So I'll be very brief about the method that we use. That's fluorescence resonance energy transfer that probably most of you here are familiar with. So we tag nucleosomes with two dyes to look at the distance changes between these 2 points of dye labeled. And the efficiency of the energy transfer has been long documented for a long time and well accepted that it's a function of distance between the two dyes, and then we can estimate the distance between two fluorophores by looking at the intensities of these two fluorophores. So that's the major method that we use. So the first system that we studied to test this hypothesis for CpG methylation was this one. This one is made of 5S rDNA sequence and xenophos levis histones that's recombinant, so it doesn't really have any modifications. And we just looked at how these termini of the nucleosomal DNA moves when we look at these individual particles. So basically, we expected to see, and if there's any dynamics in the time scale of our measurement, we would be able to see it, and by methylating it, we would be able to see any change in the flexibility of the nucleosomal DNA. Setup's pretty straightforward. We have some kind of imaging optics to flatten out the image and then the image on an EMCCD. We use TIR for excitation. The individual dots here are the Cy3 dyes that are labeled at individual nucleosomes. These are spectrally separated space, but they're spatially overlapped, but just I separated them according to the spectral wavelength of the emission. The individual dots means it's individual nucleosomes, and there are corresponding dots for Cy3 and Cy5. By measuring the intensities of those 2 points, we can estimate what are the distances between the two labels in multiple nucleosomes. All right, so we looked at the time trajectory of the distance. There might be some dynamics going on, but then we really don't have high enough resolution to figure out what are the dynamic behavior. But then we have two different populations in terms of FRET efficiency. So this FRET efficiency time trajectory tells you how the two ends of the DNA moves or getting close along time. It seems it's pretty stable. Similar, very stable, but it has different FRET. That means the distance between the two ends of the DNA is different here versus here. These two populations do exchange at a very slow rate, but they do exchange. So there's some degree of dynamics. But then if we methylate the DNA, we start seeing these short excursions to a very high FRET state. That means somehow the two ends of the DNA get very close every once in a while. So we focused on that. Before that, I wanted to briefly mention that the two different populations that we see as a FRET efficiency, two different FRET efficiency level or the populations, is probably the two different translational settings of the nucleosomes. That means there are different ways of histone core binding on a piece of DNA. If it binds in different places, it may have different hydrodynamic behavior because the DNA ends would be flappy and the fragments or the length of the fragments of the flappy end could be different when the histone core binds at different positions. And that's called nucleosome translational settings. And it's known that the 5S rDNA has two translational settings of the histone core. on the DNA. And if we anneal the nucleosomes, we see the shift in the population density in these two different translational settings. So that's what we think is happening. Nonetheless, the dynamics or the excursions to a really high FRET state is pretty significant in both cases. And we wanted to conclude that, okay, that means DNA is wrapped around the histone core more tightly when we methylate it. But then there's still this possibility that DNA ends might be completely unwrapped and then free, and then every once in a while they meet with each other to form really high FRET state. So we need to distinguish these two different possibilities from each other. So we used anisotropy measurement to estimate the flexibility of the two ends of the nucleosomal DNA. Turns out to be pretty effective because this motion, the ends of DNA, it's probably something like diffusion of fragments of DNA, 10 to 20 base pair fragments of double-stranded DNA, and that diffusion probably is in time scale of nanoseconds to microseconds, and that seems to be pretty close to the fluorescence lifetime of these dyes, so that's pretty effective. The anisotropy is pretty effective method to measure the flexibility of the termini of the nucleosome. So we used FRET anisotropy of the psi5 that's excited by the psi3, and the polarization difference or the randomization of the polarization state of the psi5 after we excite the psi3 is what we measured, and then that will tell you how flexibly the two ends of the DNA, the nucleosomal DNA, moves. This slide, I'll just go briefly. This slide is to show you that there is a one-to-one quantitative relationship between the rotational decoherence timescale of one end of DNA to the other, relative to the other, to the polarization or anisotropy of the nucleosomal DNA, psi5 emission anisotropy relative to the psi3. So if we know the fluorescence properties of the dyes like psi3 and psi5, we can measure this rotational decoherence timescale of the flappy ends of DNA by measuring the anisotropy. Setup's pretty straightforward. We just now divide the acceptor emission into two different regions or two different polarization state. And then we have one more, well, this is the raw data. So this is the donor site. This is two different polarization state of the acceptor. So now we have two-dimensional measurement. The first dimension is distance between the two, and the second dimension would be how flexible these two ends are. And we plot that. We see this high FRET state has really rigid structure. So that means it's probably the two ends of the DNA tighter wrapped around when we methylate the DNA. So that's what we tested. We could run statistics on the state lifetimes and frequency of this tight state formation, and that will lead us to this dynamics. It seems that CpG methylation lowered the energy barrier of this change, and then this change make this change or this structure somewhat more accessible when we methylate the DNA. So if that's the case, if DNA is rigidified when we methylate the DNA, that must be seen also in the internal regions of the DNA, not only the terminal regions. So that's what we tested next. So we made two different nucleosome constructs. The first one called 601 plus 39 that has these positions of labeling. And next one, 601 plus 29 that has these positions of label. And if it is true, if the tight knotting is what happens when you methylate the DNA, we will expect FRET change here to the direction where you find the FRET efficiency will decrease. Here, FRET efficiency will increase if we find that tight knotting is indeed the case upon demethylation. So these are representative traces for these four different cases. First one, 601 plus 39 unmethylated, 601 plus 39 methylated, 601 plus 29 unmethylated, 601 plus 29 methylated. So there are some dynamics like this that could be confounded by, with some dynamics of nucleosomes, but this is actually some photophysics of fluorophores, so we don't really include these kind of data in data analysis. But we use all the cocktails to suppress the blinking, photobleaching, all these things, but we still cannot get rid of these dynamics 100% of time. These things still happen. We just throw out these dynamics when we analyze the data. So this is what we saw with the first construct, that we expected FRET decrease upon the methylation if the methylation rigidifies the DNA, and that seems to be the case. Same, if the DNA rigidifies upon the methylation, the FRET efficiency increases with this construct. So that seems to be the case, and now we wonder what's the origin or nature of this change upon the methylation. And there have been some studies in the past with the cyclization dynamics, and a long time ago by Don Crother's group, that simulated Monte Carlo simulation to fit their cyclization data to two parameters. One was DNA the helical repeat length, and the second one was the flexibility. So they found upon the methylation of some repeat of CG sequences that the helical repeat increases from 10.5 to 11.0. So that's quite an increase. And then that would be interpreted as, in the nucleosomal context, this is a change. So upon the methylation, if it's elongated in double-stranded form, what you see in the nucleosomal context is it's basically tight knotting, right? With the same number of base pairs, you go further if the helical repeat length increases. And that could also, they also saw the flexibility decreases upon the methylation. And that seems to be well accepted in the field now. At least DNA becomes rigidified upon the methylation. So to monitor, to test this hypothesis, would really DNA changes its topology? It's basically topology change. And in this context, DNA topology change means the twistedness of the DNA in the double strand. Would that be changed or not? So that's the thing that we need to test. So we need to monitor the topology of the DNA in the nucleosomal context. Now, we label the fluorophores inside of the nucleosomal DNA. Now it cannot move. We anchor both sides of the fluorophores along the backbone of the DNA. So the fluorescence and anisotropy of nucleosomes are pretty high. It's at the level of 0.3334, so that's really high. And that's only probably, the small decay would be probably due to the tumbling of the entire nucleosome construct instead of local rotation of the dyes. So when we look at the fluorescence and anisotropy of the Psi5 in nucleosome, that's where the fluorophores are internally labeled. The two contributions to the decay of the anisotropy of the Psi5 would be first, the nucleosome tumbling. That's, if nucleosome tumbles in solution, that will be contributing to the anisotropy decay. Second one would be misalignment between the Psi3 and Psi5 dye. So if two dyes are misaligned, you will see some decay of the fluorescence anisotropy. So if we can measure these, we can decouple this from the convoluted effect of these two. So to measure this, we can utilize just simply excite the Psi3 and then measure the anisotropy decay of Psi3 and measure the Psi5 anisotropy decay. To measure the convoluted effect, we can just excite the Psi3 and measure the anisotropy decay from Psi5. Very similar description. What we have is this equation. If we have anisotropy decay is measured by direct excitation of the donor and acceptor together, we can decouple the effect and then measure the anisotropy decay due to the dipole misalignment, and that value can be related to the angle between the Psi3 and Psi5. So we can measure the angle between the Psi3 and Psi5. So these tell you how freely the nucleosomes tumble. And this has the information of the tumbling of nucleosome and the misalignment of the angle between these two dyes. And then we can clearly see there's difference before and after methylation. There's difference in the angle between these two. That means the gyre of upper gyre and lower gyre of nucleosomal DNA has different twistedness upon the methylation. So that seems to be fine. All work out fine. Hypothesis is still valid. So if that's the case, we should be able to see difference in the efficiency of nucleosome assembly upon the methylation. That's what we did next. Problem with this approach is under a physiological condition, you cannot expect to see nucleosomes in this strong positioning sequence called 601, disassemble or assemble. So what we used was something called NAP1, that's a protein that assembles nucleosomes by facilitating the interactions between histones and DNA. So we are focusing on DNA. So NAP1 presumably don't have any impact on the structure of DNA. So NAP1 here does just the assembly. So NAP1 just enables us to monitor the intermediate states during the nucleosome assembly under a physiological condition. So that's the role of NAP1. And this is what we did. So we label surface with a fragment of DNA, that's 601 plus some linker, labeled with a FRET fluorophore pair, and then flew in the histone octamer. It's actually not this octamer form, it's probably tetramer plus dimers. But anyways, after we elute them, inject them into the sample chamber, we would see the development of FRET signal. The instrumental setups, very similar. So we use something like millimeters fluidics. It's pretty crude. We make it with double sticky tape and it works fine. So we flew histone ESNAP1 complex together into the solution. We see the development of FRET while the DNA start wrapping up and then eventually form a canonical nucleosomal structure. Let's see. Yeah, so this is raw data. This flew in histones. You can see some development of the FRET signals. All right. As the nucleosomal DNA start wrap up, you can see the anticorrelation of the two dyes. All right, so you can just plot this intensity or intensities of the two dyes and then get an idea of how much the DNA is wrapped. So this is typically what we see when we do the experiment. From one DNA fragment, you can start seeing a low extent of wrapping and then medium extent of wrapping, that's mid-FRET state, and higher FRET state, that's almost canonical nucleosome. This lives for a long time, too long for us to measure the lifetime because the lifetime here would be limited by the photobleaching of the dyes. This is still pretty long time, but yeah, so when we analyze this type of data, we use stochastic models, like hidden Markov models. When I started writing this software package, I was hoping that I could identify some hidden states, which it turns out to be pretty ambitious. And if you can figure out new states by this way, it should be obvious to your eyes anyways. So what I use this for is reproducible data analysis. When I was in Steve Chu lab, what we did was just distribute the same set of data to 4 postdocs, and then collect the data, manually analyze data, and then average out the numbers. But I don't have 4 postdocs, I just depend on this. But anyway, so that's what we use for. But then some things sometimes obvious. Here you can see clearly anticorrelated Psi3 and Psi5 intensities, but you don't see these states are caught by the algorithm. the algorithm works in the same way as you would analyze in an exponential decay curve fitting. It's pretty much the same thing. So if you don't force this algorithm to fit this state with multiple exponential, it cannot fit everything. So this means there are more than one FRET states there. So we did manual analysis with the high FRET state. That turns out that if we don't give the dimers to the system, it lacks its canonical nucleosomal state. And if we give all the octamer components to the DNA, it forms 3 different FRET states. That's longest lived state, which is the canonical nucleosome. And this intermediate state, we put this way, was identified by Caroline Luger and Langowski group, that they labeled the histones and then saw slightly disassembled octamer with the DNA still intact. So that's probably this. We have no difference in the extent of DNA wrapping in these 2 states, but they do have 2 different structural states. So it's probably that state. We assigned this to that state. And if you can see this octamer case and tetramer case, these intermediate states are all affected by the presence of the dimers. That means, I mean, it's not known where the dimer eventually go into the nucleosome assembly path, but it seems it's clear that dimers don't recognize specific point of time to go into there and then associate themselves in the nucleosome. It's just probably randomly distributed throughout the process and just randomly bound. And at some point after tetramer somehow find its dyad position, then dimer probably have better chance to get themselves associated with both dimers and the other parts of the nucleosomal DNA. So that's probably what happens here. Strikingly, if we methylate it, these paths are all very inefficient, becomes really inefficient, and the kinetic barrier between the mid-FRET, high-FRET, so it's basically telling you the structural flexibility of the DNA becomes really low and very rigid. The DNA becomes really rigid when you methylate it. So the change in the rate is really significant. So we could conclude that the DNA methylation not only changed the structure of nucleosome, it also affects the stability of the nucleosomes kinetically. So that's kinetic effect. Nucleosomes stabilize the intermediate states or wrapped state of DNA or bent state of DNA. This is long enough time scale so we can assume some kind of thermodynamics equilibrium between these intermediate states. So we could calculate the population density and just convert them into thermodynamic stability. It seems at the highest wrapped state, the methylated DNA or nucleosome actually is a little more stable than the unmethylated DNA or nucleosome. Not a lot. This is KT, so it's not a lot that you would draw some kind of conclusion out of this. But more drastic effect, again, is the kinetic barrier. That's the flexibility or the rigidity induced upon the methylation. That seems to be the more dramatic effect. So thermodynamic effect was a StatMech study by an Italian group that they used all these dinucleotide steps and tetranucleotide steps of the crystal structures and try to calculate the effect of flexibility affecting the stability of thermodynamic stability of nucleosomes. And they found that when you have more rigid DNA, structurally more rigid DNA, to form nucleosome, you pay much less entropy cost versus flexible DNA, you have to now pay a lot higher cost for confining the conformation of the DNA into one context, that's nucleosome. Right? So that entropy effect, according to that group, dominates the thermodynamic stability of the nucleosome. That's nothing to do with the sequence-dependent nucleosome stability, so don't get confused with that. It's just for the rigidity and the flexibility. So this is a recap of the introduction for histone acetylation. So I'll be quick. So basically, the hypothesis is the histone acetylation mostly activate genes. So if we see acetylation, especially H4, that tends to be activating genes, we would expect some form of open chromatin structure. So that's the hypothesis. So H4 acetylation would induce DNA unwrapping, and that's what we wanted to test. Similar approach, we constructed two different nucleosome core particles, two different dye locations. If it unwraps directionally, this construct will show FRET decrease. This construct will show FRET increase. Seems to be the case. We also did some kind of control because we used, in this case, Piccolo NuA4 histone acetyltransferase that acetylates H4 and H2A lysins. The direction is what we expected. So the hypothesis seems to be valid when we acetylate H4 and H2A by Piccolo NuA4. We would see directional unwrapping. And the same question, what's the origin of directional unwrapping? And as I mentioned already a long time ago, when people measured the conformation of nucleosomal DNA, they found that the nucleosomal DNA has helical repeat length 10.2. That's smaller than 10.5. So this is overly twisted. So if there's any interaction between the histone and DNA that's holding this tension of over-twistedness, if we somehow release that tension, we will see topology change of the DNA again. And the charge neutralization hypothesis for lysine acetylation seems to be still well accepted. That's by acetylating the positive charge on the amine group that would interact strongly with the negative charges on the phosphate backbone of the DNA. By acetylating that positive, the lysine group will just eliminate that positive charge, and now the N-terminus group that's rich in the lysine group would lose its affinity to the phosphate backbone. And if that's something contributing to the structural integrity of the over-twistedness of the nucleosomal DNA, then we would see some opening of the nucleosomal DNA out of the nucleosome. So that's what we wanted to test. Same test. We measured the anisotropy in different formats and found that the topology of the upper gyre and the relative topology of the upper gyre to the lower gyre of the nucleosomal DNA seems to change. So that also still the hypothesis is valid. So it seems histone acetylation of H4 at least in intranucleosomal context alters its structure and opens up the nucleosomal DNA. What might be more important in histone acetylation mechanism is the internucleosomal interactions. So first, before we look at that process, we wanted to see what are the intrinsic interaction properties between the two nucleosomes without any structural constraints such as linker DNA that'll have humongous effect, in entropy effect when they interact with each other. So we use this strategy, first labeled Psi3 labeled nucleosomes on the surface, and then we eluted Psi5 labeled nucleosomes in solution and looked at the spontaneous formation of dinucleosomes. And it seems that they do form pretty stably. This is a time scale of seconds and sometimes milliseconds, hundreds of milliseconds. So they do form stable dinucleosome structure. When we acetylate the histones, this is the lifetime of these dinucleosomal structures. Some long-lived ones are probably something relevant to the high-order chromatin folding. But these short-lived ones, when I look at these things at a single molecule level, there are always these failed attempts of everything. So this is probably random encounter of these two nucleosomes to the reaction coordinate that wouldn't really lead this to anywhere productive. So that's probably this. The long-lived species is probably the ones that are relevant for high-order chromatin folding. Anyways, the both seem to be decreased by acetylating them. So acetylation of H4 inhibits the formation of the dinucleosome. When we measure the FRET efficiency, it's shifted toward to this lower FRET efficiency. That means that either the distance between the two nucleosomes gets further or the rotation will be different. But anyways, for this part, we saw that internucleosomal structure and internucleosomal interactions are affected by histone acetylation. So internucleosomally, it will induce unwrapping of nucleosomal DNA out of nucleosome. And internucleosome, it makes the separation further and inhibits the dinucleosomal formation. Of course, we have to expand this system to more physiologically relevant system. That's how this structural change would be implicated in the efficiency of chromatin remodeling. And some people believe that some portion of nucleosomes must be affected because before the any RNA polymerase pass through it. Some people think it's partial disassembly. Some people think if the positioning strength is not that big, then it would survive without any changes. So that will be the future direction. So we wanted to basically characterize the structure of the dinucleosome that's formed in a solution, because we could measure multiple different positions. So this is kind of pseudo two-dimensional FRET measurements that will give us 2 degrees of freedom in the space that with which we can characterize the dinucleosome structure. As we saw, there are multiple FRET states. This is probably nothing that's relevant to the chromatin folding. So among these two, which is more relevant to the higher order chromatin folding, we know that we need at least 1.5 to 2 millimolar level of magnesium to form chromatin, higher order chromatin folded structure. So we reduce the magnesium level to 1 millimolar to see that this population went away. So we now know that this is more relevant for higher order chromatin folding. We built a model based on the 2 parameters that we could measure, and then we characterized the model this way. So this is the model that we could characterize out of the data. 2 nanometer separation face to face, which is pretty close to what you see in the tetranucleosome crystal structure. One thing that we were surprised about was this rotation angle that's different from a crystal structure published by Richmond Group in 2004 that was tetranucleosome crystal structure that has the opposite direction of the rotation. But this direction, for one thing, explains better the previous biochemical studies of cross-linking between H4 and H2A. And the 2004 paper about the tetranucleosome crystal, they also acknowledges that the direction of the rotation should be this way. So it seems it's fine. The only exception that this cannot explain is that crystal data. That's here. So there is still a debate about how these nucleosomes compact with each other to form this 30 nanometer fiber structure. And then someone in the UK, Daniela Rhodes, made the EM images of these structures and then found that this might not be 30 nanometers. This could be 45 nanometers. And some people could have data show that it could be a mixture of these two. And it seems to be that this confirmation of the one-star helix that's the solenoid type of construction of the fiber out of the nucleosome arrays. And this is two-star helix whose basic repeating unit is 2 nucleosomes. And obviously, the confirmation would depend on conformational stability, would depend on the linker length. And lower organisms tend to have short linkers, higher organisms tend to have longer linkers. And if, for instance, So higher organisms have only a portion of genes active at a given time, while lower organisms like yeast have most of the genes active. Or in a fast developmental stage, there is an evidence, the piece of evidence that I forgot the name of the organism, but it seems faster growth phase has shorter linker length of the DNA, or NRL, or nucleosome repeat length. So if we can correlate the gene activity to one of these structures or conformations, depending on the linker length, we could correlate all these three. So for instance, if yeast that has 20 base pair linker, that's 167 NRL, if that has more active conformation of genes, and if it turns out that that adopts mostly two-star helical conformation, we can conclude that two-star helical conformation may be better for polymerases to access and have more open conformation as opposed to the one-star helix. So that's the eventual goal. We construct two different tetranucleosome arrays made of 601 sequences linked by 30 base pair linkers here. This construct would report FRET if it forms two-star helical conformations. This construct will report FRET if it forms one-star helical conformation. The first construct that was supposed to give two-star helical conformation, it does give FRET, so it forms two-star helical conformation, but then it has really well-defined substates of the structure. So there are pretty well-defined tetranucleosome structure. I don't know the motion, precisely how it's going, but there are some dynamics, and then that dynamics seem to depend on the concentration of linker histone. So linker histone is another type of histone that binds this region of nucleosome that induce this change of chromatin fiber formation. When we look at the one-star helical conformation construct, it also gives dynamic changes, but it forms one-star helical conformation. It's very dynamic, but it does form this type of structure too. And that also, the dynamics also seems to depend on the concentration of the linker histone. So first, we wanted to test if this folding behavior of tetranucleosome is something relevant to physiology. And then we measured the magnesium dependence of the structural stability of these two different conformations. And it seems that it has impact on the structural stability of the both of the conformations, one-start and two-start. So not surprising that the two-start helical conformation has slightly higher, slightly lower stability. When we use different concentration of linker histones, it seems that linker histone is the major driving force of this chromatin fiber folding. And that's pretty dramatic folding, the efficiency of folding. And only at 5 nanomolar concentration, which is about the binding constant of the linker histone. So without linker histone, even at really high magnesium concentration, the array doesn't really fold, or it's probably too dynamic to be detected with fluorescence probe that we use, which has only 10 kilohertz at most, the photon emission rate. So that's probably why. So anyways, we know that we need linker histone to fold chromatin or nucleosome array. That's currently what we do. We first wanted to characterize the structure of the tetranucleosome and how that conformation or structural stability of those conformations would depend on the linker length. That seems to be a major variable that cells utilize to control the overall activity of the genome. And the effect of H4K16 acetylation on array folding. This acetylation is known to be affecting the chromatin folding behavior. So we constructed chemically ligate H4K16 acetylated histones, and then we are currently trying to measure the efficiency of array folding with this acetylation. And also the methylation and how the methyl binding proteins would be implicated in the process of gene silencing with this mechanically altered properties of the nucleosomal DNA. This is also something I was interested in from the beginning. So there must be some form of thermal fluctuations of the structure, which is probably in this salt condition, which is about 100 millimolar level of sodium and a couple millimolar of magnesium, nanoseconds to microseconds, which is not accessible with fluorescence probes at the moment. And it's very dynamic. So I could do FCS measurement, that's what John Witham did. But that's not really single event resolution. So eventually I wanted to see if really the factor binds only when this thermal fluctuation renders this open conformation. And that could be detectable only when I use a single event resolution of the measurement. So microsecond could be accessible if use really high pumping laser with very stable dye. But this is probably too short. So I'm still thinking about doing this, but that will require some time to design the experiments. But for today, I hope that I convinced and delivered enough amount of data to convince you that these chemical modifications would impact on the physical properties of the nucleosomes and chromatin that will eventually contribute to the regulation of the gene accessibility. And single molecule methods are mostly one-dimensional measurements, two-dimensional. Three-dimensional is probably too much. So if you have a hypothesis or problem that you can reduce down to one-dimensional or two-dimensional space to explore, it could be pretty efficient tool to study such problems or test hypotheses. All right, so these guys are who worked on the nucleosome project. We are working on some properties of linker histone with Jeff Hayes at Rochester. And Ben Black is our collaborator to look at the structure of centromere-specific nucleosomes at Sandbay replacing H3. And these are funding sources, and with that, I thank you very much for your attention.
Systematic differences in the dynamics of wrapping and unwrapping nucleosomes for different DNA sequences. Get an idea of checking, learning something about stability of nucleosomes. You showed methylated versus unmethylated. It's straightforward, right?
Yeah, yeah, that's an important parameter to explore, but we haven't done anything with sequence variability.
Very interesting because.
Yeah, of course. Yeah.
Thermodynamics. You plan to do that?
We are currently looking at endogenous sequences that are still. So the problem, 601 is great. It's so homogeneous, you don't see any variability of positioning, but then that means you don't have sensitivity of changes that.
601 isn't in genomes, so who cares, right?
So to achieve the high level of sensitivity to these changes, it might be better off with endogenous sequences. So we are currently looking at some sequences. We have about 4 different sequences on hand. It's a matter of execution.
Yeah. That would be very interesting.
Yeah.
Did you ever look for any type of synergy between magnesium and H1 in the condensation process?
So far what we did was, we did use both, but then only 3 points each, so it's 9 points. So we didn't really see any synergy. It seems to be only linear in that range, so 1 to 5 millimolar magnesium. Yeah.
And it was more additive than the effect.
It's linear, linearly additive, right.
So this business about the possible intermediate states in the nucleosome formation, I mean, do you have any ideas as to what that could be? I guess you could imagine that, as the DNA wraps around the histone complex, that, there might be, because one of the questions has kind of been out that some of the work we've done is whether or not there's like a local minimum associated with the wrapping process in which the DNA has to sort of transition from a slightly bent structure to a highly bent structure. Okay, and there might be some structure that is initially only slightly bent, therefore only not well wrapped, and then it makes a transition to like a more highly bent structure. But you could also imagine that maybe it's like the wrapping of, or you mentioned the two, what are they called, gears or something? You wrap sort of almost twice around there, and so you could imagine wrapping once and then wrapping a second time.
So it's diet is heavily interacting with tetramer, and these ends tend to be interacting with one dimer and then another.
Right.
So it's probably the diet first with the tetrasome, the tetramer, so to form tetrasome first, and then dimer and then another dimer. That seems to be what salt gradient method go through. And also NAP1 also seems to function that way. So NAP1, people believe that take dimers randomly bound on nucleosome away from the DNA so that it interacts more efficiently with tetramers first. And once it forms some kind of stable structure, it'll deliver the dimers.
Right.
So yeah, it is possible that there are stable intermediates along that line, only with tetramer with presumably, I guess, a lot less wrapped or bent.
Right.
Then,
wrapped
yeah.
around the tetramer. Okay, recruits the others. Okay. Oh. Yeah.
Let's note about the distribution in solution and perhaps in the cell of tetramers versus octamers not bound to DNA. Is there a predisposition, a greater stabilization of an octamer state?
I don't think octamer is stable under physiological condition. So, yeah, it's people seem to be fine accepting that that's the structural constituent of histones. It's tetramer and dimer. Yeah, people don't seem to think that octamer exists as naked form without DNA in cells. It's only in like 2 molar, which is 2 molar sodium that's too high to make the octamer intact. Yeah. So that's our stock solution concentration. 2 molar of sodium chloride, that seems to be necessary to keep this octamer structure intact. Otherwise, they will just break down.
Okay, thank you very much.